So this is where we left off yesterday. So now that we know the difference between DNA and RNA, we can move on and address this larger topic that we call the central dogma. And the central dogma explains to us how you turn DNA into a protein. Okay? Now, that involves two processes. The first process is called transcription. And in transcription, which occurs in the nucleus, the DNA is copied and turned into mRNA. Now, remember, the DNA cannot leave the nucleus, it's protected there. So the copies or the mRNA can leave the nucleus. And we call that, of course, transcription. Now, where do those copies go? Well, when we learned about organelles, we know that ribosomes are responsible for making proteins. So those copied instructions of mRNA will go to the ribosomes in the cytoplasm to build the protein using those instructions. Okay? Any questions on that? So remember, we're saying the central dogma, that this concept is so important to our understanding of biology that it's really foundational. Okay, it's a foundational understanding in biology that we turn DNA into mRNA and then mRNA into proteins. So, how does this work? Well, transcription makes mRNA from DNA. And so the strand of DNA actually serves as the blueprint for making the RNA. So, as you see in this nucleus, you go from DNA to RNA. Now, what do you notice about the RNA in terms of its size? Is it as long as the DNA or shorter? Okay, so yeah, Kelby said shorter. It is. Because when you're creating RNA, you're not copying the whole strand of DNA. You're only copying one gene. Okay? And as we talked about yesterday, one gene codes for one protein. So the mRNA that's formed doesn't have the entire genetic code. It only copies the instructions to make one specific protein. Okay? Now, how does the RNA form? Well, it follows the base pairing rules for RNA. So if the DNA has an A on the RNA, that would be U. Okay? And you'll use the base pairing rules to form the strand of RNA. Now, cytosine and guanine don't change, they are the same. They still bond to each other, so they're still complementary. The only difference is that thymine in RNA is replaced with uracil. So if you see a strand of letters and it has a U in it for uracil, you know you're looking at RNA, not DNA. So you would know that transcription must have already happened. Okay? So an enzyme catalyzes this. So when we did DNA replication, what was the name of the enzyme that added new nucleotides to a strand of DNA? That's what split them. So which is the one that added the bases? Which one? Good. So Mitchell said DNA polymerase. Well, guess what? RNA has a polymerase too, and it's called RNA polymerase. So RNA polymerase creates a strand of DNA, or sorry, RNA using the DNA template. Sorry, I got distracted there for a second. Someone's trying to get in, so I'm going to have to end the show for a second, guys, to admit them to class. So hang on 1 second, and then we'll go back to where we were. Okay. So let me repeat that last statement one more time incorrectly. Okay. So RNA polymerase creates the strand of mRNA from the DNA template. Okay. So the RNA polymerase will only create a strand for one gene and then it stops. Okay. It doesn't read the whole strand of DNA. So what does this look like? Well, the DNA unzips, and at first you'd say like, oh, that looks like it's going to do the same thing as DNA replication, but it doesn't. Only the gene unzips, not the whole strand. Okay? So you've exposed the gene that you want to turn into a protein. Okay? And only one of the DNA strands is read. So the double-stranded DNA unzips. So now there's space between the DNA. RNA polymerase bonds to one of the strands. And then the RNA bases bond together according to the base pairing rules. Okay? So that RNA polymerase is going to create a single strand of mRNA from one side of the double-stranded DNA. Okay? Any questions on this? All right, so now we need to do a little bit of practice. So what I want you to do is answer this question. During transcription, what would be the matching mRNA sequence for the following DNA sequence? So let's say the strand of DNA has separated, the gene is exposed, and this is the gene. ATTGGCA. Okay? Now, if you turn this into a strand of mRNA, okay, what would it be? What would the nucleotides in that strand be? So take 30 seconds. I'll mute myself while you're thinking and writing, okay? And then we'll come back together and go over the answer, okay? Go. Your 20 seconds. So what bonds to T? So A stays the same. It's just the T that's here. Does that make sense? Good. So what would that, if it was DNA, what letter would that be? Let me know if it doesn't, okay? All right, so if this is our strand, okay, check your work. So A is the DNA, right? Now, typically, if we were replicating DNA, the corresponding base on this other side would be T. But we're making mRNA. So the correct sequence would be U, A, A, C, C, G, U. Okay? And I'll repeat that one more time. U, A, A, C, C, G, U. And that would be the strand of mRNA. That is the strand we would use then to make the protein at the ribosome. Okay? Now, any questions on how we got that? Okay. So, here's another example. Now, unfortunately, my lines are a little off here, but if you follow them down, right? If this is my DNA template, okay, this would be my mRNA strand corresponding to this. So A, U, G, right? C, G, U, G, U, A, U, G, C, A, U, G, C, G, C, C. Any questions on how that strand would be formed? Now, if you're doing any problems tonight and you go back and look at this, okay, you should be able to use this as an example for yourself on how to create this mRNA strand. So you might want to highlight or star this slide so that you can use it later. And of course, here's the end product then. A single strand of RNA. Now, even if you didn't know or you didn't know that this was made during transcription, if you looked at this single-stranded molecule, how could you tell that it's RNA and not DNA? There are two big things that should stand out to you. Milan? Exactly. It has uracil instead of thymine. That's one. What's the other way you can tell that this is RNA? How many strands does it have? Only one, right? And so if it is existing as a single strand, then you know that is mRNA, okay? Or RNA, okay? Now, we are going to use this strand of mRNA to create our protein, okay? So the ribosome is able to convert this mRNA, the directions for a gene in the mRNA, into a specific protein. Now, in the very first chapter of the year, biological molecules, we learned that proteins are made of what monomer? What
Amino
monomer?
acid.
Exactly. Good. Uh, was that Braylon?
Yeah.
Okay, great job. So, amino acids. So, we know that the mRNA is actually going to tell us what order to bond the amino acids in. Okay? That's what it's going to give us the information to do. So, now that we have created the mRNA, it can leave the nucleus and go to the ribosome. Remember that was our problem yesterday was we couldn't the DNA can't leave the nucleus. And proteins are made in the cytoplasm, not in the nucleus. So we've got to bridge that gap. And that's what the mRNA does. The mRNA can leave the nucleus. You can always make more. So if that mRNA does get damaged, the cell's functions are not going to be affected dramatically. It can make more mRNA. If you mess up or damage your original DNA, that's it. There's no other template to use, okay, to create these proteins. Okay? So that's going to create that would create a much more serious issue. Okay, so I'm going to have to exit again to let people in. So hang on 1 second. Okay, so hang on 1 second. Okay, so how does mRNA code for proteins? Okay, so now we've got the mRNA. How do we turn that into a protein? Well, the mRNA leaves the nucleus and goes to the ribosomes. Then the proteins are built from the instructions on the mRNA. Okay, so you want to make sure you're filling out your notes for this one. Okay, so we are able to turn the mRNA into a string of amino acids, but we need to know how. Okay, we always want to be able to explain these processes. Okay, so how do we turn this single strand of mRNA into a string of amino acids that form a protein? So, a little review. Remember that there are 20 amino acids, and they can be put together in many, really, just a limitless, a different number of orders to create different proteins. So, the order and type of amino acid determines the shape and the function of the protein. And so, if you don't bond the amino acids together correctly, the protein will have the wrong shape and it will not be able to do its job. So this process is extremely important. And it is important that it's done correctly. Now, these are the 20 amino acids. Okay? So, what I want you to think about, actually, we're going to skip this next question for a moment and we'll come back to it later. Okay? Because you're not going to be able to see both slides at the same time. So I will post this for you later and then we'll answer this as part of our homework. Okay? So we're going to leave this next slide for right now. Okay, so when you're looking at a strand of mRNA, okay, and you're going to turn that now into a string of amino acids. Now, when you look at the protein row here, you'll see that each amino acid is is abbreviated as a three-letter word. So like MET, ARG, VAL, ASN. Each one of those is an amino acid. Each one is, okay? Now, how do you go from this to this? Well, it turns out that uh, if there are 20 amino acids but only 4 DNA bases, one base cannot equal an amino acid. So, for example, if you've got 4 bases, A, U, C, and G, G cannot always equal the same amino acid because there are 20 of them. Okay? So our nucleotides are not enough. So it turns out that mRNA actually codes for proteins in what we call codons. Now, a codon is just a sequence of 3 nucleotides. So every 3 nucleotides codes for 1 amino acid. If I start with the DNA, and now I've created my mRNA. Oh, this did not show up quite correctly. So hang on 1 second. What I'm going to do is show you this on my screen instead of on this slide. This is so important. I don't want us to look at this incorrectly. So just pause for a moment, okay? And I'm going to show you on the camera now how this works. So hang on 1 second. I'll stop sharing that. And now I will share the dot cam. So we're going to ignore that for right now. All right, so back to sharing. Okay. Bear with me for a moment. Okay. So okay, I'm going to share my screen again. Okay. And now you'll see. Let's just get this set up correctly on the camera. Okay. I've got my strand of mRNA written out here. Okay. So this is my mRNA. Now, if I divide this strand up by threes, okay, each of these 3 bases is what we call a codon. And a codon just is 3 nucleotides. Now, 3 nucleotides. There we go. Now, 1 codon equals 1 amino acid. Okay. And so I drew these lines in here, not because there are lines in a strand of mRNA, because it helps me figure out where the codons are. Okay, so I took my strand of mRNA and I just divided it into groups of 3. Okay, so I counted 1, 2, 3 slash 1, 2, 3 slash 1, 2, 3 slash 1, 2, 3 slash 1, 2, 3 slash. Now, as you practice this, okay, if this way doesn't work for you, like if that doesn't help you visualize it, you can also do this. You could write each codon in a list like this. So like AUG, CGU, okay? And figure out the amino acids that way, okay? So it is up to you how you want to try to organize this, okay? This is the way that I recommend starting it, okay? Just making the slashes, okay? If that doesn't work for you, then we can figure out another way. Now, we need to use this mRNA to make a protein, okay? So, how do we figure out the order of the amino acids for the protein? Well, it used to be that you would have to memorize what the codon stood for, but you don't have to do that anymore. There is a resource that we use now called a codon chart or a codon wheel that will tell you what amino acid corresponds to a codon. And that's what I'm going to show you now. Okay? So I'll stop sharing this for a second and go back to our presentation. Oh, it's thinking. Okay, here we go. All right. So a codon equals a block of 3 mRNA bases. You can write that down. Ignore this picture because the lines are not adding up correctly. Okay. So I will have to fix this. All right. So how did I get these amino acids? Well, this is the codon chart. Now, this is how you read this. There's a chart and there's a wheel. I'm going to give you both so that you can practice both and see which one you prefer. So first, you start with the first base in the codon, or the first codon in the strand of mRNA. So the first codon was AUG. So does everyone have that in your head? AUG. So A is the first nucleotide. So I start here, A. Now the second base is U. So I've got AU. And then the third base is on this side. So AU and then G. AUG is MET or the start codon. Now, if I go back to my example, there it is, MET. Okay. Now, let's try the next one, CGU. Okay, so I start on this side. Here's the C, C, G, and then U, CGU. And look, ARG is the amino acid. Okay, this is how I find the sequence of amino acids for a strand of mRNA. So this is true for all life. And not only is it true for all life, but there are some repeats. So like, look at this right here, AUU, AUC, and AUA all correspond to the amino acid ILE, okay? Isoleucine. Now, keep in mind that the 3 letters for the amino acid, those are abbreviations, okay? It's not the full name of the amino acid. Okay. Any questions on this page right now? Now you're going to get more practice with this, but just as a concept, are there any questions about this? Okay. So, oops. So we call this translation. So DNA we transcribe into mRNA, and then we translate the mRNA into a protein. And tRNA or transfer RNA is how that happens. So the tRNA that we talked about yesterday brings the amino acids to the ribosome. Okay? Now, the way it does this is with anticodon. So you use base pairing rules yet again to figure out which anticodon in the tRNA would bond to the mRNA. Now, this part gets a little confusing, but for right now, you don't need to do this base pairing because you've got a codon wheel. So you can go directly from mRNA to the amino acids. You don't have to go through the tRNA on your papers. Now, in reality, though, we know that tRNA is what's bringing those amino acids in. So we need to address that that's how this is happening. Okay? So, turning mRNA into a protein is called translation. The instructions, of course, come from the mRNA. The ribosome is what's reading those instructions. And then the transfer RNA or tRNA is what brings the amino acids into the process. And so you can see here, tRNA is bringing amino acids in, and then those amino acids bond together. And this process continues. Okay? What you'll end up with then is a string or a chain of amino acids that bond together to form a protein. Now I'm going to pause here to give you a second to write this slide down. Okay? The blanks are in your guided notes. Okay, good. So, to summarize,
I didn't finish.
I will post these so you can copy that down later today. Okay? Oops. So, that is how, this is the story of how, or the processes for how we turn DNA into mRNA through transcription. And then we translate that mRNA into a protein. And this is how we create a protein from DNA. Now, that protein then, of course, gives us our traits. So the protein that you create for a gene impacts the traits that you show. So there are some blanks to fill in on this slide. Okay, so make sure you're filling these in now. And again, if we go too fast and you need to go back to these tonight, the slides, I will post them during fourth period today. So they will be posted for you. Okay, any questions right now? Okay, now I know this is a lot, okay? So this is where we are going to stop. Actually, one more slide and then we will stop for today. So hang on one second, okay? So then this should be your last slide that you need to fill in. Okay. So tRNA is bringing in the amino acids. Okay. mRNA is the copy of the DNA. And of course, ribosomes are the protein factories. Okay. Now, for the building, you can just write ribosome. You don't have to write all three. Like, you don't have to write ribosome protein factory. Like, as long as you know that's the ribosome, we've already memorized that um, uh, proteins are made in the ribosome. So you can just write the word there. Okay. Mhm. Oh, you didn't. Okay. So we're going to stop there for today. Now, I'm going to post these slides, like I said, during fourth period. There will also be an assignment posted today. Okay. It's a protein synthesis review. We went through so much information today. Okay. You've got to have a chance to break it down and go over it again this weekend while it's fresh in your brain. Okay. So that assignment will be due Monday. Okay. And I will post the notes later today if you need to copy them down. Okay. All right. So I am going to clean the desks now. Have a good rest of the day, everybody, and then a very nice long weekend. Oh, actually, the homework will not be due until Tuesday. I forgot we don't have class on Monday. So the homework will be due on Tuesday. Okay. Bye, everybody.